Looks like my first slide is missing, but yeah, I wanted to bring a perspective on how we can leverage material science tools, in particular electron microscopy, to the study of biological and environmental systems. And the previous speakers showed some of the amazing capabilities that we can harness at light sources, and they're really transformative. And what I would argue is that what we need are tools closer to home. We need ways to bring artificial intelligence and machine learning into our laboratories all around the world. And so that's what our team at PNNL is really focused on. We've been thinking about how to operationalize AI to aid in materials and chemical discovery, but many of the capabilities that we're building have also, I think, have cross-cutting potential for biological, for environmental research. And ultimately what it comes down to for material scientists is, of course, the atomic world, right? We'd like to harness the atomic world for emerging technologies, but to do so, we need to be able to examine atomic scale systems and larger scale systems, micron scale, nanoscale systems, macro scale systems. And so electron microscopy is one of the most powerful tools that we have to do so. In particular, electron microscopy and transmission electron microscopy allow us to examine materials all the way down to the atomic scale. They provide insight into structure, into chemistry, into composition of materials. But when we have these amazing tools that allow us to peer deeper and deeper into materials, we oftentimes struggle at generalizing it, at building information and insight across scales. And so this is where we think artificial intelligence and machine learning can really be transformative. Many of the recent advances in machine learning have looked at how we can integrate different modes of information to extract knowledge from very complex, time-evolving systems. And this is a problem that we face in electron microscopy. And so this is something that the previous speakers have already touched on, which is, how do we leverage some of these tools in material science, and what are some of the approaches that we could bring to bear on biological and environmental research? So really what we need are approaches to harness the capabilities that we're now deploying in our electron microscopes. So on the left here, I've got a plot of kind of progression of data rates. And as we think about fields such as cryo-electron microscopy, we've got advanced direct electron detectors that are generating immense rates of data, millions and millions of images that no human is ever going to be able to analyze. And the cryo-electron microscopy field has made a lot of progress in this regard. However, one of the problems that we struggle with is kind of the variety of data that we'd like to look at, the variety of features that we'd like to examine. And if you have a repeating structure, such as some kind of crystallized protein, something that you can generate in a way that is amenable to kind of imaging over and over again, you can use many of the conventional sort of analysis approaches. But in material science, in chemistry, many biological and environmental systems, you have motifs, you have structures that may be dilute, they may be rare, they may be varying, and you may not have many examples of those, yet they may determine the functionality of a system. And so using emerging machine learning techniques, we can start to integrate information at volume. We can start to harness these different data streams, parse these data streams, extract statistics from them, and then really try to build richer models and insight into these systems. Now, this is a central problem that has motivated a lot of our work. And so what I wanted to talk about today are two topics, which again are cross-cutting. So I'm going to be giving examples of material science problems, but I'm going to show how they're relevant to environmental and biological research. The first is analytics for discovery in the microscope. The microscope is a very unique problem space, and it's very different from many other types of characterization. So I'll talk about what that is. Then I'll talk about how we can start to implement that into a new workflow for end-to-end automation of the electron microscope, and how that can start to give us statistical insight into many types of materials, chemical, and biological systems. So to begin with, I think we've all kind of already heard about the challenge of machine learning, which is many machine learning approaches rely on large volumes of high-quality training data. Some of the frontiers in machine learning revolve around few-shot or zero-shot approaches, basically cases in which we don't have large libraries of labeled training data, yet we'd still like to draw some inference. We'd like to, for example, classify features, detect objects, track motion. These are canonical sort of problems in electron microscopy. This is further compounded in electron microscopy by the fact that an object can change its representation in data depending on how it's imaged. So for example, the electron beam, as many biologists will know, is very detrimental to samples, right? It will very rapidly damage them through radiolysis. In addition, depending on the orientation of an object, depending on the aberrations of our electron beam, that can change how the object is represented in the data. And so that's another problem that we deal with. And then finally, we deal with very large volumes of data. We may have images, we may have diffraction patterns, we may have spectra that are collected from a particular sample. So how do we start to make sense of that, again, absent this large volume of high quality labeled training data? So what we can do, the approach that we take, is we can use some canonical examples, some little knowledge that we may have about a particular feature, object of interest, a particular motif in our samples, and we can start to use this to discriminate and to triage data. So this approach is based on the use of a prototypical neural network that basically allows us to select canonical examples of what we care about and look at unknown regions of an image. So machine learning people love to use cats and dogs, and essentially you can use examples of these as canonical examples that a meta learner, a type of neural network, can use to discriminate unknown objects. So for example, I have an example of different types of dogs here. I can group these and just provide a few examples, and then I can compare query sets, basically unknown objects against these, and try to bin them into one of these categories. And remarkably, this is a very common scenario in the electron microscope. So here, for example, if we have electron micrographs, doesn't really matter what the particular system is here. Here we're looking at two interfaces, an interface between two materials. What we can do is we can break that down into meaningful motifs. We can subdivide that image, and then we can select regions that we may have a little bit of intuition about. So for example, this could be a known crystal structure. It could be a known defect, a known boundary in a material. In a cell, it could be a particular morphology, a particular shape. And we can use those to define what we call support sets, basically exemplars that every unknown part of the image will be compared against via a meta learner or prototypical network. What we can do is then we can segment at the chip level unknown regions of our image, and we can do this at scale. So using this little bit of intuition, we can start to make sense of very complicated data. And the important thing is we can change our task very quickly on the fly. So for example, we can separate out different regions of a material by choosing different support sets, and those support sets can change at the time of an experiment. We can separate out different types of particles. So this is a very relevant example to cellular biology, where you may want to examine different morphologies of cells. You may want to segment them from one another, distinguish them from different types of cells. And you can do this all without retraining the underlying network. The encoder that is used here to perform representation of the raw data is the same, so it's very fast and quick to train. So if we compare this to benchmarking against kind of a trivial benchmark, which is human operator labeling, this process takes forever. And it's particularly bad because it's very slow. It's not amenable to time evolving data. And so if you have intuition about the sample, and maybe your intuition changes during the labeling process, you have to restart. With this task-based approach and using a few-shot machine learning approach, you select examples of the types of objects you're interested in. The meta learner will then compare all the unknown parts of the image to those and basically assign what it thinks is the most likely candidate for those unknown parts of the image. You're going to change the task. So for example, distinguishing different types of particles, distinguishing the particles from the background, all without retraining the underlying network. This is not an incredibly sophisticated network, but it is one that is well suited to this particular problem of electron microscopy, which is limited high quality label training data. If you have that training data, you can use that to further define support sets and increase the discriminating ability of the network even further. So once you have this, the question becomes, how do you actually implement it? And this is where I would argue we've made the biggest strides. And this is a tool that we're trying to deploy more broadly, both here at the laboratory and for the community. And that's in the area of automated experimentation. So if you have the ability to make decisions and implement some kind of human-like reasoning, how do you start to deploy that on today's microscopes? This is an example of one of our microscopes here at PNNL. It's an incredibly high-end instrument, but it has 4 or 5 different control points, different detectors, different hardware made by different manufacturers. And so the challenge is pretty obvious here. You need a central controller that can actually use the reasoning, use the output of your neural network to make decisions. And then you can start to move toward high throughput screening of the kind that is very common in cryo-electron microscopy, but far less common in many types of cell biology, many types of environmental research, and certainly material science and chemistry. So what we've built, and the reference is down here, and I'd be happy to talk to anyone afterward, is a new controller, essentially taking advantage of low-level APIs that allow us to control every single part of the instrument. And we run these through an asynchronous controller that can basically issue commands and receive data at different rates. It's tightly integrated with our machine learning, so we can both acquire data in what we call an open loop fashion, where we basically sample in a predetermined manner, and then we use the results of our machine learning to more intelligently sample different parts of a particular sample, a particular grid, particular distribution of cells or objects. So what this allows us to do is this type of experiment, where you have an open loop control. Essentially, you predefine a search grid, your instrument automatically drives throughout that sample, acquires a large volume of data, then your few-shot engine is running in real time in parallel with this, and it's actually performing task-based classification. This task can change during the experiment. So maybe at the start, you just want to find all the particles on the background. Maybe as your experiment goes on, you want to separate different types of particles. You can change that on the fly. And then you can assign feature maps and you can assign coordinates for the features of interest. And this, I think, is one of the most potentially impactful areas. You can start to adaptively sample those regions, right? One of the big challenges, again, is mitigating dose in the electron microscope, particularly in biological systems. If you can put your dose exactly where it's needed, you can get the information you want without damaging the underlying structure. So then we think that this is a powerful platform for end-to-end discovery. We can load samples into our instrument. We can automatically identify sample landmarks. There's some other work going on here that I'm not talking about today, but it's covered in the paper, and I'd be happy to talk more about it. We can unwrap distortions and correct the orientation of our sample. Here, you're just looking at a grid, a distribution of particles on the grid. The instrument can automatically acquire montages from thousands, tens of thousands of particles. We can then use our few-shot engine to basically classify, again, by task, what objects we may be interested in, determine their coordinates, and then generate rich statistical distributions based on task. So the point here, and the most important thing, is that this is generalizable, right? This can move from one task to another very quickly, and the underlying engine can be ported from one microscope to another. So we've already deployed it here on 2 instruments. This is available for any JOL instrument, which is one of the most commonly used electron microscopes. And we think it has tremendous potential for biological and environmentalists. So with that, I wanted to acknowledge the teams, a very large team here at PNNL, students at the University of Washington, SULI interns, SCGSR interns, and others who made this work possible. And it's really, the convergence of the material science, data science, chemists that are making this possible. So with that, I'd be happy to take any questions. And I wanted to acknowledge Office of Science as well as PNNL LDRD for supporting different aspects of this work. Thank you.
Thank you so much, Stephen. Do we have any questions? You gave us a great taste of the communication and controlling all different data sources to one thing. That was very relevant also to the first invited speaker talk this morning, right? Jake he also made that message that, yeah, not just to randomly generate data and then try to put them together. So yes, this needs to be in a controlled fashion, and we all know that, but do we do it? Sometimes we need some work on this, yes. Do we have any questions in particular?
Yes. So you talked a lot about the capacity for this learning approach to apply to synthetic biology. Are there any, how should I put this? Have you applied it to biological systems yet? Or do you have research in progress where you plan to apply this to biological systems? And if so, I'd appreciate if you could talk about that.
Absolutely. So, I mean, certainly Alice and I have talked about that. There are some examples that I haven't shown here where we are starting to apply it to cellular imaging, but we haven't done much. And so I think that's part of the conversation here, right? I'm trying to show tools that I think are relevant, and have shown success in other domains. And I'd be very interested to learn more about some of the biological applications, but it's something that's underway.
Yeah.
Right. Thank you very much.
And yeah, thank you very much. But needless to say, we know that material science is, a generation beyond and above the biological applications. So yeah, we are just behind you, but we'll get there with your help. Thank you very much for your talk.